All right. Thank you for joining us today. Reminder that you are all muted and participating in listen-only mode. We are allowing time at the end of the three speakers' presentations for questions. Please feel free to ask questions in the Q&A box at any time. I will now turn it over to you, Dr. Rizzo.
Thank you, Katie. Welcome to today's Congressional Neuroscience Caucus briefing on COVID-19 and the brain. I'm Matt Rizzo. I'm the chair of neurology at the University of Nebraska Medical Center, and I'm also the chair of the American Brain Coalition. Today, our coalition, together with the American Academy of Neurology, the American College of Neuropsychopharmacology, and the Society for Neuroscience are partnering with the Congressional Neuroscience Caucus. And we're hosting this briefing today on emerging research on neurological and psychological effects of the SARS-CoV-2 virus. We're very happy to have the caucus with us. It's co-chaired by Congressman Earl Blumenauer and Congresswoman Kathy McMorris-Rodgers. It's been going for decades, since 2010. The intent is to build awareness on the intrinsic role that brain research plays in understanding ourselves and society, communicating progress and benefits of the research, and promoting changes in policy that are critical to supporting this research. So we're really, really honored today to have Congressman Blumenauer. He represents the 3rd Congressional District in Oregon. He's a lifelong resident of Portland, and he was first elected to the House of Representatives in 1996. Time flies when you're having fun, I guess. And he's used his time there on the Ways and Means Committee to promote many aspects of healthcare access and environmental reform. He's been a tireless champion for patients suffering from neurological and psychiatric disorders, and research to develop cures to reduce the impact of these disorders. So together with Congresswoman McMorris-Rodgers, he serves as the co-chair, as I mentioned, of the Congressional Neuroscience Caucus, and we're really, really grateful for his leadership on these important issues. So pleased to meet you, Representative Blumenauer, and please take it away.
Thank you, Dr. Rizzo. And I will be brief because we have 3 very impressive speakers who can help us deal with yet another critical area of neuroscience dealing with contemporary problems. And we're going to see that this is woven throughout the challenges with COVID-19. As scientists and clinicians learn more about this novel coronavirus, evidence suggests ways it may affect the brain in a number of different ways. They've observed patients with COVID-19 experiencing confusion, stroke, seizure, or loss of smell. Many of these symptoms and complications are also found in other types of infections, and we need to be able to have more precise information. We don't know whether these are the result of a virus, an immune response, or something else altogether. We don't know what the long-term effects of COVID-19 might be, but evidence from other viruses can give us a clue, as Dr. Nath will discuss. Additional physical isolation, which is being experienced across the country, the uncertainty regarding the virus, the economic slowdown has led to worsening mental health. It's clear we need to invest further research, being able to provide mental health support. And ultimately, it's clear we need to invest in further research into the COVID-19 to have a better understanding of how this disease affects the brains. We must assure that we include communities of color in this effort, as has been documented time and time again in ways that are really, I think, unsettling to anybody, how deeply the country is divided in terms of the disproportionate impact on people of color, particularly African Americans, Latinx. This is part of the overall challenge. I could not be more pleased that the Neuroscience Caucus is able to partner with you to bring this important discussion in a virtual sense. I hope in the not too distant future, we'll be able to gather again on Capitol Hill in not have to be too socially distant, and be able to be engaged in these conversations. But in the meantime, I look forward to this conversation today and welcome you to the Caucus briefing.
Thank you so much, Congressman Blumenauer, and thanks for setting the stage for our three speakers. They are quite accomplished, and they are running the gamut from sensory deficits to cognitive to emotional to social, in line with what your opening comments addressed. And our first speaker today will be Dr. Sandeep Robert Datta, goes by Bob. He's Associate Professor of Neurobiology at Harvard Medical School, and he has an amazing pedigree. He got his MD and PhD from Harvard. He worked as a postdoctoral fellow at Columbia University and joined the Harvard Medical School Department of Neurobiology in 2009. And his lab focuses on sensory cues, particularly odors, how they're processed by the nervous system, how the brain uses this information to process salient cues. And he received the NIH New Innovator Award, a Burroughs Wellcome Career Award in the Medical Sciences, an Alfred P. Sloan Research Fellowship, Searle Scholars Award, a Valley Young Investigator Award, a McKnight Endowment Fund Scholar Award, and he's been named a Fellow of the National Academy of Science Kavli Scholars Program. That's really impressive. Bob, we'll turn it over to you now, and look forward to learning something.
Thank you, Dr. Rizzo. Can everyone see my screen? It's okay? Okay, so again, my name is Bob Datta. I'm a neurobiologist at the Harvard Medical School. And before I begin, I'd like to thank everyone for making the time to consider the impact that COVID-19 is having on the brain. It's clear that millions of people across the globe are going to be infected with the novel coronavirus. And it's equally clear that a substantial fraction of those people will have changes in neurological function. And so this is a subject that's, I think, well worth all of our attention. I thank you for your time. A main, if not the main, neurological manifestation of COVID-19 are changes in the sense of smell and taste. So smell and taste are chemical senses. They allow you to interact with chemicals in the forms of odor chemicals and scents, and molecules that are in food that you encounter when you eat. And the chemical senses, essentially allow you to interact with the chemical world. I'll just briefly remind you a little bit about the chemical senses. Of course, we smell through our nose, which houses a structure called the olfactory epithelium. And that epithelium includes sensory neurons that interact with odors in the environment. These neurons send structures called axons that actually poke little holes in the bottom of your skull. And these axons then terminate in a structure in your brain called the olfactory bulb, which is responsible for your brain's ability to process odors. Taste, of course, occurs through taste buds in your tongue. And there's this third chemical sense called kinesthesis, which is responsible for your ability to detect spicy food or to feel the cool in mint gum. Now, current research suggests that COVID-19 actually attacks all three of these chemical senses independently. That being said, we know the most about how the novel coronavirus might attack smell, and it's the predominant thing that COVID-19 patients complain of when it comes to chemical sensation. So I'm going to limit my comments to smell. So when the pandemic began, there nearly immediately began to be anecdotal reports of people losing their sense of smell and food tasting funny. And that prompted really a global effort to try to understand the degree to which changes in smell and taste might be linked to the novel coronavirus. That has led to efforts like this. Here on the left is shown an app that was developed by researchers in the United States and in England that has been downloaded now by more than 2 million people. And it tracks both whether or not you're coronavirus positive or negative and COVID symptoms. An analysis of data from this app and many million individuals has revealed that upwards of 90% of people who are COVID positive complain of some change in their sense of smell and taste. Now, I want to stress that our understanding of this fraction, the number of COVID patients that have a change in smell or taste, is still emerging, in part because mostly people are reporting to us how they think their smell and taste is working, as opposed to us as researchers taking objective measurements of their sense of smell and taste. Those data will emerge, and I rather expect that these preliminary results will hold up. It's, I think, important to note that smell loss can be the first thing a patient notices when they have the coronavirus. And in about 25% of patients, it's actually the only symptom they ever get. Despite the fact that that might make you think that they have a mild case of COVID-19, those patients that only complain of smell loss are in fact infectious and can transmit the virus to others. And because of the prominent association between changes in smell, taste, and COVID, the CDC and WHO have both included these kinds of chemical sense changes on their list of COVID-19 associated symptoms. Now, the fact that smell, taste, and COVID are so closely related has led researchers to hypothesize that you might be able to use the sense of smell or taste to predict whether an individual actually has COVID or not. And that is bolstered by a recent study showing that recent loss of smell is more predictive of someone having infection with COVID than other symptoms that are typically associated with other viruses, like fever or cough or shortness of breath. So based on these and other data, there's really a race to develop practical tests for smell that might be used for screening individuals in the community to assess whether or not they might have COVID. So one version of this has been recently developed by my friend and colleague Mark Albers at the Massachusetts General Hospital here in Boston. And as you can see on the right, this is just a scratch and sniff test. And the idea is that these cards will be distributed to the community. Individuals would be able to scratch individual odors, and then through a web-based form, report back the quality and intensity of all of the odors on the card. And Dr. Albers and his colleagues are developing algorithms that, based on these data, might be able to predict whether or not you have COVID. An interesting complementary approach has been generated by my friend and colleague Noam Sobel at the Weizmann Institute. So here, they proposed a model in which individuals go to their kitchen cabinets and actually identify 5 objects that have strong smells. Here I have picked Nutella, peanut butter, vinegar, cumin, and toothpaste. And they ask individuals on a daily basis to report back how they're feeling and the quality and intensity of these smells that people just pluck out of their kitchen. Dr. Sobel and his colleagues are also developing algorithms that might be able to use this kind of kitchen-based approach to allow individuals across all sorts of communities to be able to accurately predict whether or not they've been infected with a novel coronavirus. And I want to stress that these kinds of approaches are extremely important from a public health perspective, because obviously there are limits to the availability of PCR-based testing in many communities and across the globe. And this kind of kitchen-based test might be an important complement to standard clinical tests and might enable us to isolate individuals who are likely to spread the disease. Now, this all raises the question of how does this novel coronavirus actually attack chemical sensation? And you might imagine, as we did when we first started thinking about this problem, that the novel coronavirus is just like the common cold on steroids. It's a really bad cold. But I want to stress that what we know about the natural history of this new virus suggests that it's really nothing like the common cold. As I'm sure all of you have experienced, when you get the cold, you get a stuffy nose that is accompanied by a ton of inflammation that prevents odors from getting back into your olfactory epithelia and prevents odor sensation. And when the stuffiness clears up, you get your sense of smell back in a matter of days. In addition, we know that in the common cold, there are a small number of individuals whose olfactory epithelia are damaged by cold-causing viruses. And recovery from that, from the loss of smell due to viral insults like that, can take months to years. For the coronavirus, many patients report they lose their sense of smell almost like a switch has been turned off in the absence of a runny nose or any obvious inflammation. And typically the sense of smell returns in a couple weeks, not days or years as occurs when you get anosmic with the common cold. And all of this suggests that whatever the SARS coronavirus is doing, it's doing something different than the typical cold-causing viruses. So my lab and many other labs have investigated possible mechanisms through which this novel coronavirus might attack smell. And I'm not going to take you through all of the details of this slide, I just want to remind you of what I told you earlier in my talk, which is that inside your nose, there's this structure called the olfactory epithelium that houses sensory neurons here, which are colored red, that have these little dendritic cilia that are in the air space of your nose and detect odors. And these neurons have structures called axons that puncture the base of your skull and then go up into a structure called your olfactory bulb, where they interact with neurons that process information about smells. We've been able to use the tools and tricks of modern molecular biology to ask which cells in the olfactory epithelium and the olfactory bulb might be susceptible to the coronavirus infection. And what we've discovered has surprised us. We think in our preliminary analysis that the olfactory sensory neurons that are the business end of the epithelium and responsible for smell might not be directly targeted by this virus. Instead, it looks like in the olfactory epithelium, the support cells that nourish these olfactory sensory neurons might be the primary target. In addition, when we look in the olfactory bulb, the neurons that make up the brain circuits that process odor information don't seem like they're likely to be infected by the virus. Instead, vascular cells that are responsible for delivering blood and therefore oxygen to the olfactory bulb, these vascular cells might be infected. And so the story that's emerging, again, through our preliminary work, is that support cells in both the olfactory epithelium and the olfactory bulb may be the primary targets of this novel coronavirus. And frankly, that's pretty good news. It means that in most cases, the virus is unlikely to be altering the key neurons that are responsible for odor processing. This might enable most patients ultimately to recover. I want to take a moment at the end here just to mention something about those patients that may take longer to recover, that have persistent anosmia. And I want to remind you that turns out to be very important from a clinical perspective. We as humans are visual creatures, and we're used to thinking of smell as a kind of aesthetic sense or a bonus sense that doesn't really matter very much. But the truth is that our sense of smell is intimately related to our own sense of emotional well-being. And I want to explain very briefly why. If we think about our sense of sight, light falls onto our retina, and information from our eyes and our retinas is transmitted through a chain of neurons, many, many neurons, before that information arrives at a part of your brain called the amygdala, which is responsible for your emotions and for allowing you to have a positive emotional response to something you see. In contrast, there is one neuron that connects your nose to your amygdala. There's an incredibly intimate neural relationship between your ability to smell things in your nose and how those things make you feel. And that's because smell is your most ancient sense and was thought to evolve first, and is thought to be very closely related to neural processes like attraction or avoidance. And what that means is that your sense of smell really is responsible in a very day-to-day way that you don't really notice for giving you a sense of emotional well-being. And we know this because people who have chronically lost their sense of smell actually have significantly higher rates of depression, anxiety, and suicide. And that's on top of the other risks you have when you lose your sense of smell, like not eating food and having a poor nutritional status, and not being able to detect danger odors like those emitted from fire or natural gas. So I think, although most patients with this novel coronavirus are going to recover probably in a matter of weeks, there's going to be a fraction of people whose recovery takes a long time and may not ever occur. And I think it's important that we pay attention to those patients as this pandemic evolves as well. So just to end and to highlight the main takeaways, I just want to remind you that anosmia is a major consequence, a neurological consequence of this COVID-19 syndrome. And my hope is that we can use our understanding of how the virus attacks the olfactory system as a model for better understanding in general how the virus might affect brain function. I think the sense of smell might be very useful for large-scale community screening for whether or not you're COVID positive. I think that's a very exciting avenue of future work. I think there's a ton of research that really remains to be done, particularly in humans, about how chemical senses are targeted by this virus and how we're going to go about potentially curing people who have debilitating losses in their chemical senses as a consequence of this pandemic. That's work that's really important, that has to be ongoing. And finally, I hope I've convinced you that COVID-19 related anosmia may have significant public health impacts and something we should pay attention to going forward. I thank you all very much for your attention.
Thank you so much, Dr. Datta. And I think the audience will recognize that he made very complex information simple. knowing that everybody in intelligence have good brains, but not everyone has the technical expertise to understand the scientific details. But now we understand a lot because of this talk. Now for a broader view of the nervous system and the effects of COVID, I want to introduce Dr. Avindra Nath. He's the chief of the sections of infections of the nervous system and the clinical director of the National Institute of Neurological Disorders and Stroke. He got his MD from Christian College in India in 1981, residency from University of Texas Health Science Center in Houston, fellowship in multiple sclerosis and neurovirology there, and then a fellowship in neuroaids at NINDS. He's held a variety of faculty positions in Manitoba and University of Kentucky. In 2002, he joined Johns Hopkins as a professor in neurology and the director of the Division of Neuroimmunology and Neurological Infections. He joined NIH almost a decade ago in 2011. He's a clinical director of NINDS, the director of the Translational Neuroscience Center, and the chief of the section of infections of the nervous system. And his research broadly focuses on the pathology of retroviral infections of the nervous system and developing new diagnostic and treatment approaches for these diseases. So thank you so much, Dr. Nath. Take it away.
Okay. Thank you, Dr. Rizzo, for that very kind introduction. And also wanted to thank the Caucus for the invitation and to the audience for participating in this presentation and the discussion later on. So I'm going to talk about the neurological complications of coronaviruses. So this is much more broader than just COVID-19, although the bulk of the talk will be related to COVID-19. So there are a number of coronaviruses that infect humans. There's a huge variety of coronaviruses that can infect other mammals. And but there are a total of 7 now that have been identified, 6 that are listed here, and plus now the COVID-19 or SARS-CoV-2. And of these human coronaviruses, you can divide them into alpha and beta coronaviruses. And all the beta coronaviruses, all 4 of them here listed, have been shown to cause neurological complications. So the neurological complications can broadly be divided into 2. You have the para-infectious manifestations, which is during the time when the viral syndrome is occurring. You can have a number of manifestations, and then after the viral symptoms disappear, some neurological manifestations can occur, which we call the post-viral syndromes, and most of them are immune-mediated. So listed here on this inverted triangle are in order of prevalence for each of these complications. And just as Dr. Detak mentioned that anosmia can occur in the vast majority of the patients, it is the most common symptom. And then inflammation of the muscle or muscle aches, also very common. The brain can get involved either due to multi-organ involvement or directly due to strokes. I'll talk a fair bit about this in just a bit. And very rarely you can get meningitis or encephalitis, and remains an area of controversy whether the respiratory nuclei within the brainstem can be directly involved or not. With regards to post-viral syndromes, again, the brain can get involved, the spinal cord, the nerves, and also a number of interesting immune syndromes, Kawasaki disease and ME/CFS may also occur in these patients. So a number of these human coronaviruses have been shown to cause encephalitis. Here with SARS-CoV-2, again, you can see some inflammation here in the temporal lobe, but there are isolated case reports. And in this one, they could find the virus in the spinal fluid. Again, this is a single case report with another coronavirus of a child who had immune deficiency. And then the SARS, which is the closest to the current SARS-CoV-2, here they looked at 8 patients in the brain, and all 8 of them, they could find viral RNA in the neurons in the hippocampus, suggesting clearly that at least these viruses have the ability to invade the brain. And Dr. Dada explained this about anosmia, and he mentioned that to date we don't have conclusive evidence that the virus can enter through the nose, but this is an interesting animal model, again with a murine coronavirus, where they do inject the virus into the nose, and they can show here over a period of days, so 1, 2, 3, 4, you can see this green fluorescence tracking, and it can track all the way down to the brainstem. So within the brainstem, there are these nuclei that are just compacted in small region that really control the respiratory system. And so an area of investigation currently is to determine if there is invasion of these nuclei, either by the virus, viral products, or by the immune system. Then with regards to post-viral syndromes, here with MERS coronavirus, people showed that a number of patients would develop acute disseminated encephalomyelitis. Acute means over a short period of time, disseminated, that means you get patchy involvement of a large part of the brain as well as the spinal cord. And so you can see here in the MRI scan, there's a lot of this high intensity signal, this white stuff that you see here is all abnormal. And again, in this second patient here, you can see from day 5 to day 28, a progression of these symptoms in this patient with more and more involvement of the brain. And here they suggested that this was either an encephalitis or it could also be a vasculopathy, because you see these punctate involvement here. Then there's a single case report coming from Detroit, and where they showed this one patient who had COVID-19 and developed this acute necrotizing hemorrhagic encephalitis. So there was bleeding in the brain. All this dark stuff that you see here is blood, and it's in the thalamus of the brain. And you can see here on another image the extent of the edema and swelling that's there. And that is classical bilaterally symmetrical thalamic lesions with this entity. And most often that is thought to be mediated by cytokine release. And then of all the CNS manifestations, I think strokes are the most common that are seen. And there are a wide variety of strokes that are being described in this population. And it can be due to occlusion of the venous system, and they can present with strokes or sometimes with what we call as benign intracranial hypertension. They can be arterial involvement, and they can be multiple arteries can be involved at the same time. And then you can see small blood vessel involvement too. So there's some data that was recently published through Mount Sinai showing that micro thrombi were present in the brain of a number of patients. The underlying pathophysiology is not entirely clear. I think it is becoming more and more common that coagulopathies are occurring in these patients. Why that may be the case is not, still remains unknown. Few patients with antibodies against phospholipids have been reported, and that's usually present in patients who, in the non-infected population, you often see that with lupus and other kinds of diseases of that sort. And so it is interesting that antiphospholipid antibodies would be present in this patient population. And then, of course, if there's damage to the heart, which can occur from COVID, then there's a possibility that clots can go from there. So individuals who have underlying vascular risk factors such as diabetes, hypertension, that are greater risk of developing strokes, and also those individuals who have respiratory involvement or multi-organ involvement. So here's an example of these microhemorrhages. All these dark spots that you see is blood leaking into the brain. And you can see all throughout the brain with lots of microhemorrhages here. And then, as I said, you can get multiple arterial thrombi, and here's the middle cerebral artery distribution on one side, and then a little branch of that on the opposite side. So you can see that there's acute infarcts on both sides of the brain. And then these are some autopsy specimens that we have received in our lab, and we're just starting to look at them. But you can see here very closely very clearly here that in the pons, there is a hemorrhage right here. And this is another part of the brain. Again, you can see some bleeding here. And you can see a lot of these blood vessels are very congested. Yeah? So, and in fact, we've looked at now over a dozen brains. Almost all of them have congestion of the blood vessels, and some of them have microhemorrhages. And the other thing we have the ability to do at NIH is to do very high resolution MRI scans on postmortem tissue. And so here's a slice of the pons, and we put it through a 11.7 Tesla magnet, making about 100 micron a section. So in order to do that, it takes about 36 hours just to do an MRI scan of this slice here. But it provides very interesting information because you can see these areas that look abnormal, all these white areas, and there are a whole slew of these that you can see throughout the pons. And that is very important because this is the area where you would expect the respiratory nuclei to be present. And so now we are going to be doing histology on these tissues to try and understand what the basis of this abnormal signal is. And this is like from another patient from the frontal lobe, and you can see again a lot of congested blood vessels, and you can see all this fluffy stuff here that is all abnormal. And this is a slide I borrowed from a neuropathologist at University of Iowa, and Dr. Hefty. And he looked at the olfactory bulb and as well as the medulla. And he shows that you can see in both areas, there's a lot of microglial cell activation. And so these are cells that are equivalent of tissue macrophages in the brain. And if they are activated, that suggests that the innate immune responses have been activated. So there's certainly some abnormalities at both sites within the brain. So what are we doing at NIH right now? NIH has an extramural component and the intramural. I'm as part of the intramural program. But the extramural program is in charge of providing research funds for various programs. And they are currently providing supplements to individuals who already have grants through the NIH to study COVID and the nervous system. And they developed a coordinating center in order to collect a huge database on these patients who are complaining of neurological symptoms. And they are looking into clinical trials of thrombotic agents for treatment of stroke of patients with COVID. Within our own program, we're putting together cohorts of patients that will undergo deep phenotyping and will be followed from time of acute infection to determine if they're long-term sequelae. As I showed you, we're characterizing the neuropathology of the infection, and we're developing humanized in vitro models to study the pathophysiology. But a lot of unanswered questions still remain. We do not know what the long-term sequelae are. We very barely understand the pathophysiology. We do not have treatments for most of the neurological complications. We do not know what happens to patients who already have underlying neurological disorders. If they now develop COVID, what are the consequences? We are very hopeful there will be vaccines, but what we do not know is the vaccines themselves could have neurological complications or immune-mediated complications. If the virus does, sure enough, the vaccine could too. And then if we are developing treatments, we can't forget the brain. We need to make sure that we have treatments that can also protect the brain. And another thing that we really need to think about is that we do not have enough BSL-3 labs or autopsy suites in the country. It came to me as a big surprise. But we haven't faced an epidemic of this nature. And so conducting autopsies has been a major limitation. And drug development has been a major limitation because you don't have BSL-3 labs where infected virus can be cultured and studied. So we need to develop that kind of infrastructure to be able to address these issues. If access to healthcare is very limited for individuals currently, and long-term monitoring is absolutely critical. So I'd like to acknowledge a few people. Here are the neuropathologists with whom I'm working with closely. There are 2 people in my lab who are doing the pathology and all the PCR. This is the MRI group, and Walter Korshas is the director of our institute, and the other 2 are part of our extramural program. So I'll stop here.
Thank you so much, Dr. Nath. You've expanded the dimensions beyond what Dr. Datta launched. We went from smell to a variety of disorders of the brain, and as you point out, they can be immune, they can be vascular from stroke or hemorrhage. Maybe there can be long-term sequelae, even degenerative. Who knows? You know, maybe Parkinson's or Alzheimer's will be at a greater risk in people who have COVID-19 or not. We don't know. And you also raised the important issues of we need to have following patients, we need to have access to tissue, we need to have registries. So these are all important issues that are emerging out of our talks. I want to mention that Dr. Emily Troyer is going to speak next. And she got her MD from University of Toledo in Ohio, trained in residency in general psychiatry at University of Illinois at Chicago, and finished in 2017. She was a chief resident there. She moved to do a child and adolescent psychiatry fellowship at UCSD, and she's been the chief fellow there. She's been an NIMH T32 postdoctoral fellow in the Department of Psychiatry, and she's currently investigating the roles of host-immune and gut microbiome relationships in pediatric onset OCD. She's had mentorship from Dr. David Rosenberg, Dr. Bob Knight, and Dr. Susie Hong. So currently, she and her colleagues are looking at potential neuropsychiatric sequelae of the new coronavirus. So Dr. Troyer, please take it away.
Okay, thank you, Dr. Rizzo, and good afternoon, everyone. First of all, I want to say thank you to the Congressional Neuroscience Caucus and the sponsoring organizations and all of the attendees for being with us today. And like you said, my name is Dr. Emily Troyer, and I'm honored to be here with my esteemed colleagues today. And let's see here. Okay, so in April, my colleagues and I published a review in Brain Behavior and Immunity where we explored potential neuropsychiatric symptoms in the context of the novel coronavirus pandemic. And I'd like to acknowledge my co-authors here, Dr. Susie Hong and Dr. Jordan Cohn. And in the Hong Laboratory at UCSD, we focus on understanding neuroendocrine and cellular inflammatory mechanisms as they relate to neuropsychiatric and neurodegenerative outcomes. Today, I'll be providing a broad overview of neuropsychiatric symptoms, either already described in the COVID-19 pandemic or speculated to be processes for which monitoring should be vigilant based on data from past viral outbreaks. And for those interested, I've shown here the Journal of Neurology, Neurosurgery, and Psychiatry is maintaining a regularly updated repository of all publications related to such outcomes, so this is a helpful resource. And throughout this talk, I'll be using the term neuropsychiatric to refer to psychiatric symptoms that arise in the context of an illness with the capacity to influence the central nervous system, such as COVID-19. And as I know this caucus is aware, neurologic and psychiatric disorders are brain disorders. And at times, the differentiation between the two can be a little bit confusing. But as a psychiatrist by training, today I'll be focusing my talk primarily on symptoms related to cognition, to mood, to perception, and to behavior. And while I don't want to minimize the potential deleterious effects of the pandemic on public mental health in our society at large, I will just be focusing on potential symptoms in individuals infected with the novel coronavirus. And I'd also like to discuss the brain and COVID-19 through a developmental or lifespan perspective lens, and I'll share my thoughts on potential implications of that. And finally, I'll share my thoughts on how we move forward as a medical and scientific community, and also as a society. So before we discuss COVID-19, I'd like to give a brief historical context for the group. Epidemiologic evidence from past viral outbreaks has demonstrated that neuropsychiatric symptoms can accompany acute infection or can follow it by weeks to decades in recovered patients. And a salient example of this is an epidemic of encephalitis that occurred around the beginning of the last century, alongside the last major influenza pandemic. And we refer to this as encephalitis lethargica or von Economos encephalitis. The acute manifestations were diverse. They typically included delirium, which is a confusional state, hypersomnolence or excessive sleepiness, and cranial nerve palsies, typically paresis of muscles that move the eyes. But acute symptoms could also include psychosis, catatonia, abnormal movements, and others. And chronic manifestations typically included depressive and Parkinsonian symptoms. In fact, it's estimated that in the decades following this epidemic, up to half of all Parkinson's disease in the population was post-encephalitic. And it's important to mention that we still don't know the exact cause of encephalitis lethargica. We don't think it was a direct effect of the influenza virus per se, but that it was probably a post-infectious immune-mediated process akin to what we now recognize as autoimmune encephalitis, but this is speculative. I think the most important take-home message from this, though, is that while the influenza pandemic of 1918 was devastating for several years, most of the disability and social and economic burden in the following decades was associated with neuropsychiatric symptomatology. So what do we know about neuropsychiatric symptoms in COVID-19? Listed here is neurologic syndromes that have been reported in the literature, and I won't discuss these in detail because Dr. Nath eloquently spoke to this topic, but I think this list demonstrates the diversity of what clinicians are currently seeing. And over time, we're also starting to see reports emerge of psychiatric symptoms in COVID-19. And listed here are symptoms that have been reported at least once in the literature so far, and I won't go into detail about all of them, but I will discuss a little bit more the bolded ones, namely delirium, neurocognitive impairment, depression, post-traumatic stress, and psychosis or schizophrenia. So in terms of cognition, delirium, like I said before, is a confusional state. It's not uncommonly experienced by individuals hospitalized with a critical illness. And delirium is an independent risk factor for mortality and functional and cognitive decline after hospitalization. And the first bullet point here refers to a recent meta-analysis by Rogers et al. They pooled studies of all neuropsychiatric symptoms in patients hospitalized for severe infection during the past coronavirus outbreaks, namely SARS and MERS. And they showed that almost 30% of patients hospitalized with severe illness had confusion, and about 19% still had memory impairment at long-term follow-up, which was an average of 3 years after the acute illness. At the same time, almost a quarter had not returned to work. And right now, the exact prevalence of delirium in COVID-19 isn't known, but it's currently being reported in 9 to 65% of COVID-positive patients. And these data are coming from studies in various contexts, including of nursing home patients, of patients seen in the ED, and hospitalized and critically ill patients. So the question remains, since delirium and neurocognitive decline following ICU hospitalization are not unique to COVID-19, we need to know to what degree COVID-19 may or may not pose additional risk for these over and above what would be expected of an infectious illness or ICU stay. In terms of mood symptoms, again, I'll reference the meta-analysis by Rogers et al., which demonstrated that of patients hospitalized for severe SARS or MERS, about a third had depressed mood during the initial hospitalization, and the post-illness point prevalence of depression was about 15%. We don't have a lot of data in COVID-19, but there is one prospective study of hospitalized patients with mild to moderate COVID-related pneumonia from Italy that demonstrates almost 40% of patients report depressed mood. Now, pictured on the right here is a graphic from a recent article in the Washington Post, which reported data from the US Census Bureau, showing that about a third of all Americans are currently reporting symptoms of depression or anxiety. So we will need robust epidemiologic studies over time to understand if COVID-19 infection is an independent risk factor for depression over and above the psychological distress that's currently being experienced by most of the country. And in regards to trauma symptoms, this meta-analysis, again by Rogers et al., demonstrated that after severe SARS or MERS, 32.2% met full criteria for post-traumatic stress disorder, again at a mean follow-up of 3 years after the illness. And we don't have good data about this so far in COVID, but there is a study from China of 714 clinically stable COVID-19 patients preparing for hospital discharge, and an overwhelming majority of them report at least some trauma-related symptoms. And they define that as a score of at least 50 on a civilian PTSD symptom checklist, the PCLC. And this alone doesn't constitute a diagnosis of PTSD, but certainly warrants further evaluation. Post-ICU PTSD is a recognized entity outside of COVID-19, and recent meta-analysis demonstrated the prevalence of this is about 19 to 22%. So given the data that we have from SARS and MERS, it may very well be that COVID-19 poses independent risk for PTSD over and above what would be expected in a critical illness. Now, psychosis is defined as a disconnection from reality-based thinking, frequently manifesting as hallucinations or delusions. It can be seen in a number of neuropsychiatric syndromes, including delirium and dementia. The psychiatric disorder most commonly associated with psychosis is schizophrenia, and epidemiologic evidence dating back to influenza outbreaks during the last century suggests a possible link between viral infection and schizophrenia risk, in that incidence of schizophrenia was increased in the decades following influenza outbreaks. So again, the first bullet refers to meta-analysis data from past coronavirus pandemics. And in COVID-19, we're already seeing several case reports and case series of brief or isolated symptoms of psychosis during COVID-19 infection. And a study from China in preprint suggests that they're seeing about a 10% increase in incidence of outpatient schizophrenia diagnoses, which they noticed in January 2020 compared to the previous 3 Januaries from 2017 to 2019. And they noticed this was particularly in individuals aged 39 to 50, which would be older than what would be expected for this type of diagnosis. Further, last week, the first case report of post-COVID anti-NMDA receptor encephalitis was published, and psychosis is a common feature of this disorder. So I'd like to switch gears a little bit here. So far, we've discussed neuropsychiatric symptoms reported during or up to 3 years after coronavirus infections, but epidemiologic data from the last century suggests the incidence of schizophrenia, for example, was increased in the decades following the outbreaks, particularly amongst individuals who were in utero during the outbreak. And the maternal immune activation hypothesis, which is depicted here, may be one mechanism by which such phenomenon could occur. And in this model, individuals exposed to inflammation in utero, regardless of the cause, whether it be infectious, autoimmune, parental stress, etc., these individuals might be at increased risk for neurodevelopmental disorders such as autism spectrum disorder or schizophrenia later in life. And while there's no current evidence to suggest in utero exposure to COVID-19 is associated with adverse birth outcomes, long-term monitoring of these children and their neurodevelopment would certainly be prudent given what we know about maternal immune activation. Another consideration is that even after birth, different types of neurodevelopmental processes are ongoing through adulthood. So exposure to infection or inflammation might have different outcomes depending on the age of the exposure. And we saw in the case of encephalitis, lethargica, for example, that prepubertal onset was associated with more hyperactive or hyperkinetic presentations, while postpubertal onset was associated with more hypoactive presentations like depression or Parkinsonism. And this is, to further illustrate this point, this is data from a preprint from a UK-wide surveillance study, CoroNerve. And again, this hasn't yet been peer reviewed, but the authors demonstrate that in the first 153 cases, the distribution of individuals affected by neuropsychiatric symptoms of cardiovascular etiology is different from that of individuals affected by altered mental status of non-cardiovascular origin. And so you can see here that altered mental status of non-cardiovascular origin is observed in a relatively younger age distribution. So to summarize, I've gone through a whirlwind overview of some neuropsychiatric sequelae for which individuals infected by COVID-19 might be at heightened risk, including neurocognitive, mood, trauma-related, and neurodevelopmental disorders. I've discussed COVID-19 in the brain through the lens of developmental and lifespan perspectives, which suggests that the full extent to which COVID-19 could impact incidence of neuropsychiatric disorders may not be fully appreciated for several decades. And so I think moving forward, we definitely need to engage in longitudinal prospective monitoring of individuals infected with COVID-19 from in utero through advanced aging. We need to understand the neuroimmune mechanisms involved with these outcomes. We need to understand if we're facing new syndromes or if we're facing syndromes that we know at an unprecedented scale. And we need to continue to build capacity to mitigate the neuropsychiatric burden in the population. As clinicians and scientists, I think we need to work to break down the false dichotomy between physical and mental health so we can understand shared disease mechanisms and potential treatments. And we need to continue to work toward decreasing stigma and improving parity so that individuals affected can get the services that they'll need to thrive. So with that, I will close.
Thank you so much, Dr. Troyer. Thanks, Dr. Datt, and thanks, Dr. Nath. We have a few moments for questions. We solicited some before the webinar, and we received some subsequently. I'm going to toss one to you, Dr. Datta, first. It's a pragmatic question. We got pragmatic, theoretical questions, some are across the lifespan, some related to acute presentations. But can one use smell as a surrogate for temperature for screening?
That's a terrific question. The way that we imagine, I think for the most part, using smell to screen is longitudinally. So if you know you're COVID negative, you can go into your kitchen and smell some things and then watch your own sense of smell change. And the reason why the longitudinal aspect is important here is because, as I'm sure many of you are aware, your sense of smell is really personal and individualized. Just in the way that some people are colorblind, there's a huge amount of variation in the smells that different individuals can detect and can't detect. So there's some thought of using smell as a kind of screen more generally. For example, there are police departments in South America that actually have been carrying sniffing sticks with them to screen their risks in interacting with people they pull over in cars. But I think the data really need to emerge to let us know whether those kinds of screens in general will be substitutes for temperature taking, or whether or not this kind of technology is better suited to longitudinal monitoring in a home setting. But it's a great question.
Thank you so much. I'm going to throw the next question to Dr. Nath, and it's a little bit technical and also longitudinal. So SARS-CoV-2 binds angiotensin converting enzyme 2 receptors, and these are located in a part of the brain called the posterior cingulate. It's a place where you get pathology like plaques and tangles in Alzheimer's disease, and also maybe the substantia nigra, which is implicated in Parkinson's disease. Is it possible that COVID-19 will lead to neurodegenerative disease in patients who contract the disorder?
So it's a very good question. We've looked at that data, And there's one paper published from China that claims that, and they analyzed existing databases on RNA expression in various areas of the brain and compared it to other things. We feel that the jury is still out. The number of cells that they're detecting are very, very few, and the number of techniques and other things used in these various databases are very different. So I don't think at this point one can really conclude that we have enough of the ACE2 expression in the brain and that there are regional differences. But I think these are important questions that we really need to study further.
Thank you so much. I want to mention another question which might go to Dr. Troyer, but I don't know that we have a chance to fully address it. But it has to do with suicide and intimate partner violence in this pandemic and preventive efforts. And I think probably there's not enough time to answer that. There are other critical social issues like food insecurity of kids who are not making it to school and rely on school lunch and so on and so forth, along the lines of the healthcare disparities that Congressman Blumenauer mentioned at the beginning. So I think we're going to wrap up now. I want to say that we are extremely grateful to have maybe 150 staffers and ABC members and other neuroscience research supporters on the call today. And we have, for the staffers, a few requests. And the first one is to assure robust and sustainable funding increases for biomedical research everywhere that it happens. And that can be the NIH, the CDC, Defense, the VA, and so on. The other thing I think we can extract from today's talks is the need to follow what happens in COVID-19 and create registries of surviving patients, and also of people who test positive and maybe don't even develop the disease that we know of, so that we can track their future health better and assess long-term implications like the ones that Dr. Nath mentioned. Thirdly, I think it's pretty important for us to have biospecimens and tissue samples, and you even heard about autopsies from Dr. Nath. There's a lot of important data and lessons that we'll lose if we don't have access to this tissue. I want to mention also that if your boss is not a member of the Congressional Neuroscience Caucus, please have him get in touch with Mr. Blumenauer or Ms. McMorris-Rogers on the floor or in committee, because I think this is a really important endeavor for you to be involved in. So we're entering a new normal after this pandemic. We have to understand the effects of SARS-CoV-2 on the brain. And I want to thank Drs. Dada, Nath, and Troyer for helping us a lot to do that with their insights. And thanks also to the Congressional Neuroscience Caucus. Thanks for partnering with the ABC, the American Academy of Neurology, the American College of Neuropsychopharmacology, and the Society for Neuroscience. And special thanks again to Congressman Blumenauer for joining us today. Thank you, everybody. It's been great.